Welcome to Learn While Sleeping. This is the podcast where you're able to fall asleep, rest, forget about your day's stress, and at the same time, manage to learn while you're doing so. There's no exam or test, nothing you have to prepare for or try hard for, so don't worry and relax. If you like this format, feel free to check out on Apple Podcasts or Spotify. We also have our YouTube channel. Thank you, and enjoy the show. Take a slow breath. Tonight, we begin with the molecule that defines every living thing. DNA. Deoxyribonucleic acid. It's more than just a code. It's a physical structure carefully designed by chemistry to store information with perfect precision. Picture two long chains, twisted around one another in a graceful spiral. This spiral is the double helix, the iconic shape of DNA. Each chain is made up of repeating subunits called nucleotides. Every nucleotide has three parts: a phosphate group, a 5-carbon sugar called deoxyribose, and one of 4 nitrogenous bases: adenine, thymine, guanine, or cytosine. The sugar and phosphate form the backbone of the molecule, like the rails of the ladder, while the bases reach inward to form the rungs. If you've listened to any of the other episodes, this is not a completely new topic. We have discussed this many times, and now we're going to go a little bit more in depth. Adenine, or A, always pairs with thymine, or T. Guanine, or G, always pairs with cytosine, also known as C. These pairs are held together by hydrogen bonds. 2 bonds between A and T, and 3 between G and C. Because of this pairing rule, each strand is the complement of the other. When we say the complement, it means that if one strand reads A, T, G, C, the opposite strand will always read T, A, C, G, the complete opposite pair. This complementarity is the reason DNA can be copied with such accuracy. Each strand acts as a template for the other during replication. Now, imagine looking more closely at the helix itself. Each turn of the spiral contains about 10.5 base pairs. This regular twisting creates 2 grooves along the surface: a major groove and a minor groove. Proteins that interact with DNA, like transcription factors and polymerases, well, they recognize specific patterns of atoms in these grooves. They read the information not by breaking bonds, but by sensing the shape of and the chemistry of the exposed edges of the bases. That's how the cell reads the genetic code, not by unzipping it completely, but by touching it lightly, where the information peeks through the grooves. Let's review that in your mind for a moment. DNA is made of nucleotides. The bases pair with A with T and G with C. The strands run anti-parallel, one from the 5' to the 3' direction, the other to the 3' to the 5' direction. Hydrogen bonds connect bases. The sugar-phosphate backbone holds the structure. And finally, the double helix completes one turn every 10.5 base pairs, forming major and minor grooves. These features together make DNA both stable and readable, strong enough to protect genetic information, yet flexible enough for enzymes to access it. In the next part, we'll look deeper into the forces that hold DNA together, like hydrogen bonding, base stacking, and how salt and pH influence the molecule's stability. You'll see why DNA can survive for thousands of years, yet still unwind easily inside a living cell. We've seen the structure of DNA, the double helix of two complementary strands. Now, let's explore why this structure is so stable. What keeps those strands from falling apart, and yet allows them to separate exactly when the cell needs them to? Between each base pair, tiny attractions called hydrogen bonds hold the strands together. Hydrogen bonds form between a slightly positive hydrogen atom and a slightly negative nitrogen or oxygen atom. In DNA, they occur between specific atoms on the bases. 2 bonds between A and T, and 3 bonds between G and C. Those extra bonds make GC pairs slightly stronger and harder to break. That's why a DNA region rich in G and C bases has a higher melting temperature, or TM, than one rich in A and T. Hydrogen bonds give DNA both specificity and reversibility. They're strong enough to maintain pairing, but weak enough to let the strands unzip during replication or transcription. If the bonds were much stronger, DNA would never open. If they were weaker, genetic information would just fall apart. Life depends on this perfect middle ground. The second force that stabilizes the helix is what's known as base stacking. Base stacking is actually considered to be much more important when it comes to stabilizing the helix. The nitrogenous bases are flat, aromatic rings. They are hydrophobic surfaces that avoid water. Inside the helix, the bases stack tightly on one top of another, minimizing their contact with the surrounding solvent. These stacked rings attract each other through van der Waals forces, tiny electrical interactions between an atom, each weak on its own, but together, the cumulative effect of millions of stacked bases creates powerful stability. You can try imagining it like a column of coins. Each coin will press lightly on the next. Individually, they're fragile, but together, they're solid. This is the essence of base stacking. Another key factor that affects DNA stability is the salt concentration of its environment. The phosphate groups on the sugar-phosphate backbone are negatively charged. Because of this, the two strands naturally repel each other. However, positively charged ions in the surrounding solution, for example, sodium or magnesium 2+, they will neutralize that repulsion. When salt concentration is high, these ions shield the negative charges, allowing the strands to stay closer together. As a result, DNA becomes more stable, and the melting temperature, or TM, increases. When salt concentration is low, the repulsion between phosphates is stronger, and the DNA strands separate more easily, which will then lower the TM. So, in the lab, scientists carefully control salt levels when performing experiments like hybridization or PCR, also known as polymerase chain reaction, so as to maintain precise DNA behavior. We also should discuss the effect of pH. pH also influences stability. At a neutral pH, the hydrogen bonds between bases remain intact. But in alkaline conditions, like high pH, well, the hydrogen bonds will then break, and DNA denatures into single strands. At acidic pH, something different happens. The glycosidic bonds between bases and sugars can then hydrolyze, meaning that they create apurinic sites and they'll damage the molecule permanently. This is why DNA is most stable in near neutral conditions. It's essential for that gentle balance to be found inside living cells. Let's take a quick mental review. Take a moment to picture the helix in your mind. 2 strands twisting together anti-parallel and complementary, held by 3 forces working in harmony. First force, hydrogen bonds, connecting complementary bases like adenine and thymine, which will then have 2 hydrogen bonds, and guanine and cytosine, which will then have 3 hydrogen bonds. The second force is base stacking, hydrophobic interactions and van der Waals forces that stabilize the core. And the third force is ionic shielding, positive ions that neutralize the negatively charged backbone with the salt concentration. All 3 of these forces depend on pH, salt, and the base composition. And all 3 together explain why DNA can remain intact for thousands of years, yet unwind instantly when a cell divides. Now that we understand what holds DNA together, let's see what happens when those forces are broken. In the next part, we'll explore how DNA strands separate and reform, the phenomena of denaturation and renaturation. You'll learn how scientists use these processes to measure stability, to calculate melting temperature, TM, and to detect DNA using light. Until now, we've looked at the forces that keep the double helix together. But life and molecular biology, they depend just as much on the fact that those forces can be broken and then restored. When DNA strands separate, the process is called denaturation, or melting. When they find each other again and reform the helix, it's called renaturation, or annealing. These two reversible processes lie at the heart of replication, PCR, southern blots, and every hybridization-based experiment in modern molecular biology. As the temperature of a DNA solution rises, the hydrogen bonds between complementary bases begin to weaken. Eventually, at a specific range of temperatures, the two strands fully separate. This is the melting point, or TM. This exact point is the temperature at which half of the double-stranded DNA has become single-stranded. The TM depends on several variables. The base composition, which is the GC-rich DNA that has more hydrogen bonds and higher TM. Secondly, the salt concentration, because higher ionic strength stabilizes the backbone and raises the TM. And finally, the length of DNA. Longer strands require more energy to denature, which would just mean a higher TM. There's even an empirical formula to estimate it for short sequences. For quick lab estimates of oligonucleotide primers, a simpler version is used for this formula, and it's basically as follows. The TM will roughly equal 4 degrees Celsius multiplied by the guanine plus the cytosine content plus 2 degrees Celsius multiplied by the adenine plus the thymine content. These equations aren't just math, they let scientists design primers that will anneal at exactly the right temperature for PCR or hybridization. As DNA melts, something remarkable happens. Its ability to absorb ultraviolet light increases. At 260 nanometers, single-stranded DNA absorbs about 30 to 40% more UV light than the same DNA when double-stranded. This jump in absorbance is called the hyperchromic shift. Why? In double-stranded DNA, base stacking limits how much UV light each base can absorb. When the strands separate, the bases are exposed to solution, free to absorb more light. By monitoring absorbance at 260 nanometers while gradually heating a sample, scientists can track DNA melting in real time and then pinpoint the TM from the middle of the curve. This is one of the most simplest and elegant measurements in molecular biology. If melted DNA is cooled slowly, complementary strands will meet again, and then realign through base pairing. Thus, the double helix will reform. This process, called renaturation or reannealing, depends on both time, concentration, and temperature. Why do they depend on these three things? Well, for time, the strands must have a chance to collide, so they need enough time. For concentration, more DNA molecules means more possible encounters, so higher concentration is important. Finally, temperature. If it's too cold and random mispairing occurs, and then if it's too hot, well then pairing can't begin. There is a sweet spot, usually about 20 to 25 degrees Celsius below the TM, where correct hydrogen bonding happens most frequently. The reversibility of denaturation and renaturation is essential. It allows DNA to be manipulated over and over without permanent damage. The same principles make DNA hybridization possible. When denatured DNA from two different sources is mixed and cooled, complementary regions can base pair, forming hybrid DNA molecules. If the sequences match perfectly, the hybrid is stable. If there are mismatches, it melts at a lower temperature. This sensitivity lets researchers detect genetic similarity or mutation. In southern blots, a labeled DNA probe hybridizes to its complementary sequence on a membrane. In polymerase chain reaction, short primers anneal to their target sequence before being extended by DNA polymerase. In microarrays, thousands of probes on a chip capture matching strands to measure gene expression. Each technique relies on the same simple principle you just learned: heat to separate and cooling to reform. Let these key ideas settle in. First, denaturation, which is strands that separate when hydrogen bonds break. Renaturation, which is strands that rejoin when complementary bases align. Melting temperature, it's the point where half of DNA is single-stranded. The hyperchromic effect, which is increased UV absorbance at 260 nanometers when DNA melts. And finally, hybridization, the pairing between complementary strands from any source. Every modern DNA-based method depends on these same thermodynamic truths. In the next section, we'll leave DNA behind for a moment and explore its more flexible cousin, RNA. You'll see how one more oxygen atom and one different base change everything about how life reads its own instructions. DNA is the library of life. It's stable, protected, carefully preserved in the nucleus. But a library can't act on its own. It needs a messenger to carry the words out into the world. That messenger is RNA, or ribonucleic acid. Though it shares much of DNA structure, it behaves in completely different ways. And those differences are the reason that life can function. At first glance, RNA seems nearly identical to DNA. It's also a chain of nucleotides connected by phosphodiester bonds. But two small differences change everything. Firstly, the sugar. In RNA, the sugar is ribose, not deoxyribose. Ribose has an extra hydroxyl group, or an OH, on the 2' carbon of the sugar ring. That single oxygen atom makes RNA more reactive and less stable than DNA. It's more prone to hydrolysis, which is why RNA rarely survives long in cells, or in lab experiments, without RNase inhibitors. The second difference is the base composition. Instead of thymine, RNA uses the base uracil, or U. Uracil pairs with adenine through 2 hydrogen bonds, just like thymine, but it lacks the extra methyl group that makes thymine slightly more hydrophobic. Together, these differences give RNA a completely different personality. DNA is stable and permanent. RNA is temporary and versatile. Like DNA, each RNA strand has a 5' to 3' direction, meaning nucleotides are always added to the 3' hydroxyl end. But unlike DNA, RNA is single-stranded. This means it doesn't have a permanent complementary partner to pair with. Instead, RNA folds back on itself, forming short, double-stranded regions wherever complementary bases happen to align. These local pairings create secondary structures, such as stem loops or hairpins, which are regions of paired bases connected by unpaired loops. Also bulges, where a few unpaired nucleotides disrupt the pairing. And finally, pseudoknots. These are complex 3D tangles formed when bases far apart in sequence pair with each other. Each of these shapes give RNA unique functions and binding sites for proteins, small molecules, or other RNAs. In addition to the standard Watson-Crick base pairs like AU and GC, RNA can form non-standard pairs like GU wobble pairs. In a GU, hydrogen bonding occurs between the oxygen and the nitrogen atoms in a slightly shifted alignment. Through the less stable than guanine-cytosine pair, it's still strong enough to maintain structure. This wobble adds flexibility to RNA's folding and to the genetic code itself. It is what allows some tRNAs to recognize more than one codon. Because of these alternative pairings, RNA structures are less rigid than DNA helices, yet often more intricate. When RNA does form double helical regions, its structure is slightly different from DNA's. The presence of that 2' hydroxyl group pushes the sugar in a completely different conformation, an A-form helix rather than DNA's B form. An A form is more compact, with about 11 base pairs per turn and a deeper, narrower major groove. This difference affects how proteins interact with RNA. Most protein-RNA interactions occur in specific pockets or loops, not by reading along the major groove as they do with DNA. Let's talk about the main types of RNA. There are 3 essential types of RNA found in all cells. Messenger RNA. This is the copy of a gene that carries instructions from DNA to a ribosome. It determines the sequence of amino acids in a protein. Then there's transfer RNA or tRNA. This is a small and folded into a cloverleaf shape. tRNA carries amino acids to the ribosome. It then matches them to the correct codon on the mRNA during translation. Finally, ribosomal RNA or rRNA, the structural and catalytic core of ribosomes. Large complexes that synthesize proteins. rRNA doesn't just provide structure, it performs the actual chemical catalysis of peptide bond formation. Together, these RNAs coordinate the process of gene expression, turning the information in DNA into functional molecules. RNA is less stable than DNA. That's why it serves as a temporary copy rather than a permanent archive. But that fragility is also in its strength. It allows cells to control gene expression quickly, turning certain genes on and off in response to changes in the environment. In viruses like influenza, coronavirus, or HIV, RNA itself serves as the genetic material, proof that life doesn't need DNA to evolve and replicate. So, even though RNA is short-lived, it is the molecule of action, translating the stillness of the genome into the movement of life. DNA remembers, RNA responds. Together, they create a rhythm, a pulse, that turns the genetic blueprint into the living cell. In the next section, we'll go even deeper, beyond RNA as a messenger, into RNA as a catalyst. You'll learn how RNA molecules can behave like enzymes, cutting and joining other RNA strands, and how that discovery reshaped our understanding of life's origins. For decades, scientists believed that only proteins could act as enzymes, that catalytic power required the complexity of amino acid chains. RNA was thought to be a mere messenger, a courier that carried DNA's instructions. Then, something unexpected happened. In the early 1980s, researchers discovered that RNA itself could catalyze reactions. These molecules, called ribozymes, changed everything. They revealed that RNA is not just a passive copy of genetic code, but an active reactive molecule, capable of shaping and rearranging itself. Well then, what makes RNA catalytic? The secret lies in RNA's structure. The 2' hydroxyl group on its ribose sugar makes RNA chemically versatile. That OH can donate or accept protons. It can act as a nucleophile, and help form transient covalent intermediates. All the tricks of a true enzyme. Combine that with RNA's ability to fold into complex three-dimensional shapes, like stems, loops, bulges, and pseudoknots, and you get a molecule that can build active sites much like a protein does. So while DNA is designed for stability, RNA is designed for reactivity. RNase P is the first ribozyme that was discovered. One of the first earliest examples was RNase P. This enzyme's job is to trim extra nucleotides from the 5' ends of tRNA molecules, producing mature tRNAs ready for translation. RNase P is composed of both RNA and protein, but experiments showed that the RNA alone could perform the catalytic reaction. The protein only assists with stability. This discovery proved that RNA can behave like an enzyme completely on its own. It was the first direct evidence that nucleic acids could be both information and function. Soon after, scientists found another kind of ribozyme, self-splicing introns. These are RNA sequences that can cut themselves out of longer RNA transcripts without any proteins. The process works through a reaction called transesterification. The 2' hydroxyl of a specific adenine within the intron attacks the phosphodiester bond at the exon-intron boundary. This breaks the RNA backbone and forms a loop structure known as a lariat. A second reaction rejoins the 2 exons, releasing the intron. In simple terms, the RNA edits itself, removing what it doesn't need and joining what it does. Another fascinating ribozyme is the hammerhead ribozyme, first discovered in plant pathogens called viroids, small circular RNAs that infect plants. The hammerhead ribozyme catalyzes a precise self-cleavage reaction, cutting long RNA strands into smaller functional units. Its structure resembles a 3-bladed hammerhead, 3 short stems radiating from a central catalytic core. Within that core, specific bases and magnesium ions create a tiny chemical micromachine, positioning a 2' hydroxyl group to attack a neighboring phosphate and break the strand. It's incredibly small, around 40 nucleotides, yet perfectly folded to do its job. How do these ribozymes work? All ribozymes share a few essential catalytic strategies. Firstly, general acid-base catalysis, where RNA bases or bound metal ions help shuttle protons. Then there's metal ion catalysis. Usually Mg2+ ions or magnesium ions will stabilize the negative charge of the transition state. Then there's proximity and orientation. Folding brings reactive groups close together in just the right geometry. These are the same tricks used by protein enzymes, proving that complex catalysis doesn't require amino acids, only the right shape and chemistry. These discoveries gave rise to a profound idea, the RNA world hypothesis. Imagine a time billions of years ago, before proteins, before DNA. The early Earth was filled with simple organic molecules, and among them, perhaps, were strands of RNA. RNA could store information, just like DNA, but it could also catalyze chemical reactions, like enzymes. That means a single molecule type could replicate itself, evolve, and adapt. The very definition of life. Over time, proteins likely took over most catalytic roles because their 20 amino acids offer greater chemical diversity. DNA, more stable than RNA, became the preferred information storage molecule. But the spark, the first self-copying system, may have belonged to RNA. Even today, the ribosome, the machine that builds every protein in your body, is itself a massive ribozyme. The active site that forms peptide bonds is made of rRNA, not protein. Every time a cell synthesizes a protein, you're witnessing a remnant of that ancient RNA world still at work. Let's review these key ideas before you rest deeper. Ribozymes are RNA molecules with enzymatic activity. RNase P and self-splicing introns prove that RNA alone can catalyze reactions. Hammerhead ribozyme is a small self-cleaving RNA found in viroids. RNA uses 2' hydroxyl chemistry, metal ion catalysis, and precise folding for activity. The RNA world hypothesis suggests early life was based entirely on RNA. The modern ribosome still carries that legacy as an RNA-based catalyst. So, DNA is not just a messenger, it's a survivor from the dawn of life. Flexible, reactive, creative, the molecule that once might have ruled the Earth. In the next section, we'll move from the natural chemistry of molecules to the laboratory tools humans have built to study and reshape them. You'll learn about phosphatases, nucleases, and the remarkable precision of restriction enzymes, the molecular scissors of modern biology. In nature, enzymes shape and repair DNA and RNA constantly. In the laboratory, scientists have learned to borrow these same enzymes and to use them as tools to explore and re-engineer life's code. Each tool has a specific purpose. Some cut DNA, some join it, and others modify it to prepare for cloning or sequencing. Let's explore them one by one. Phosphatases are enzymes that remove phosphate groups from molecules. In cloning, this is used for a clever reason. When scientists cut a plasmid vector open to insert a new gene, the vector's ends naturally want to rejoin themselves, a process called self-ligation. To prevent that, the vector is treated with a phosphatase. By removing the 5' phosphate groups, the vector can no longer seal its own ends. Only when a foreign DNA fragment with intact 5' phosphates is inserted can the ligase enzyme join them together. So phosphatases act as safety guards. They ensure the plasmid doesn't close without the gene of interest inside. They are like the deactivators. Nucleases are the cutters. They are enzymes that break phosphodiester bonds, the links between nucleotides in a DNA or RNA chain. There are two main types of nucleases: endonucleases, which cut within a strand, and exonucleases, which remove nucleotides from the ends. Each has a specific role. Some nucleases act randomly, digesting nucleic acids into fragments. Others are highly sequence specific, and those are the ones that revolutionize molecular biology. Restriction endonucleases are nature's scissors. Restriction enzymes are a special class of endonucleases originally discovered in bacteria. Their natural role is defense. They recognize and cut up foreign DNA, such as that from viruses. Each restriction enzyme recognizes a specific DNA sequence, usually 4 to 8 base pairs long. These sequences are often palindromic, meaning that they read the same forward and backwards on opposite strands. When the enzyme finds its target, it cuts both strands of the DNA. Some make blunt ends, cutting across straight. Others create sticky ends, cutting in a staggered fashion and leaving short single-stranded overhangs. Those sticky ends are extremely useful. They can easily pair with matching overhangs from other DNA fragments. It's like puzzle pieces that fit together only in the correct orientation. Then there are the types of restriction enzymes. There are several types: type 1, type 2, and type 3. But type 2 restriction enzymes are the ones used in research. Type 1 and 3 recognize specific sequences, but cut at variable distances away from them. Type 2 enzymes, however, cut exactly at their recognition site. They are simple, reliable, and precise, perfect for cloning and mapping DNA. Then there are isoschizomers and neoschizomers. Some enzymes recognize the same DNA sequence but behave differently. Isoschizomers are enzymes that recognize the same sequence and cut it in the same way. For example, HPL and MSPL both recognize CCGG. Neoschizomers are enzymes that recognize the same sequence but cut at different positions. For example, AATLL and the enzyme ZRAL. Some enzymes are even sensitive to methylation, which is how bacterial cells protect their own DNA from being cut. If a recognition site is methylated, certain restriction enzymes will not cleave it, an elegant natural safeguard. Let's talk about the compatibility and sticky ends. Not all restriction enzymes cuts are compatible with one another. Only fragments with matching overhangs can be joined. For instance, an enzyme that leaves a 5' overhang ending in AA and TT can only be joined to another fragment with a complementary TT and AA end. When cloning, researchers choose enzymes carefully to create compatible sticky ends for ligation, the process of joining fragments back together. Ligase is the glue. Once a fragment is inserted into a plasmid vector, The two pieces must be sealed into one continuous molecule. This is done by DNA ligase. Ligase forms phosphodiester bonds between adjacent nucleotides, repairing breaks in the sugar phosphate backbone. In nature, ligases fix DNA during replication and repair. In the lab, they finalize recombinant DNA molecules. So if restriction enzymes cut and phosphatases prevent religation, ligases complete the puzzle, joining vector and insert into a single functional plasmid. Before sequencing becomes widespread, scientists use restriction mapping to determine the layout of DNA fragments. By digesting a piece of DNA with different combinations of restriction enzymes and comparing the sizes of resulting fragments on an agarose gel, they could infer the positions of the enzyme sites. It was like solving a jigsaw puzzle using only the pieces' lengths, a foundational skill in molecular biology. Let's consolidate the key tools you've learned so far. Phosphatases remove phosphate groups to prevent unwanted ligation. Nucleases are enzymes that cut DNA or RNA. Restriction endonucleases are precise DNA cutters used for cloning and mapping. Ligase seals DNA fragments together. And finally, restriction mapping determines site positions based on fragment lengths. Each of these enzymes has a place in the delicate choreography of cloning, like cutting, modifying, joining, and verifying DNA. In the next part, we'll build upon these foundations and explore how they became together in cloning and sequencing, how scientists create recombinant DNA, screen for successful clones, and read the very letters of the genetic code. A vector is a carrier DNA molecule that can replicate inside a whole cell. The most common vectors are plasmids, small circular DNA molecules found naturally in bacteria. A good plasmid vector has three essential features: an origin of replication, or an ORI for short, so that it can copy itself inside the cell. A selectable marker is the second thing, such as an antibiotic-resistant gene, so only cells that contain the plasmid will survive. And the third feature is a multiple cloning site, or MCS. This is a short region containing many unique restriction enzyme recognition sites, allowing scientists to insert DNA fragments at a precise location. Step 2 is to cut and insert the gene. Using restriction enzymes, both the plasmid vector and the gene of interest are cut at specific sites, usually with enzymes that create compatible sticky ends. The cut gene fragment is then mixed with the opened plasmid, and DNA ligase is added. Ligase seals the sugar phosphate backbones, creating a single circular recombinant plasmid, a hybrid molecule of natural and foreign DNA. This recombinant plasmid is then introduced into the bacteria, often E. coli, through a process called transformation, where cells take up the DNA from their surroundings. Each bacterium that successfully takes up the plasmid will multiply, forming a colony. Each colony presenting millions of identical copies of the recombinant DNA. Step 3 is preventing self-ligation. Before ligation, plasmids are often treated with phosphatases, as you learned earlier, to remove 5' phosphate groups and prevent the plasmid from closing without an insert. Only DNA fragments that supply their own phosphatases can be joined, ensuring that ligation happens only when the insert is present. This step increases the success rate of cloning dramatically. Step 4 is blue-white screening. After transformation, how do scientists know which colonies contain the gene insert? For that, they use blue-white screening, a simple yet elegant genetic trick. In this plasmid, the multiple cloning site is placed inside a gene called LAC-Z, which encodes an enzyme called beta-galactosidase. This enzyme can break down a compound called X-Gau, producing a bright blue pigment. If the plasmid does not contain an inserted DNA fragment, the LAC-Z gene remains intact, and the bacterial colony turns blue on X-Gau plates. If the plasmid does contain a foreign DNA insert, the insert disrupts the LAC-Z gene, the enzyme is not made, and the colonies stay white. So, under the soft life of a lab incubator, a scientist can simply look at a plate, blue colonies for empty plasmids, white for successful recombinants. It's molecular biology's way of color-coding success. Even after blue-white screening, we have step 5, which is verification. Scientists verify their results using restriction mapping or DNA sequencing. Restriction mapping involves cutting the plasmid again with specific enzymes and then checking the sizes of resulting fragments on an agarose gel. The pattern of bands reveals whether the insert is the correct size and orientation. But to truly confirm identity, sequencing is the gold standard. There are several methods of sequencing, each building on the last. The first is Maxam-Gilbert sequencing, or the chemical method. Developed in the 1970s, this method relied on chemical cleavage of DNA at specific bases. DNA fragments were radioactively labeled, treated with base-specific chemicals, and then separated by polyacrylamide gel electrophoresis. By comparing fragment lengths, scientists could read the sequence. But the process was time-consuming and involved toxic reagents. It served as an important foundation, but it was soon replaced. The second method is Sanger sequencing, or the dideoxy method. Frederick Sanger introduced a more elegant method in 1977, known as chain termination sequencing. It used DNA polymerase to synthesize a complementary strand, starting from a primer bound to a single-stranded DNA template. The reaction mixture contains normal nucleotides, or DNTPs. and then a small proportion of dideoxynucleotides, or DDNTPs, small special molecules lacking the 3' hydroxyl group. When a DDNTP is incorporated, the chain stops growing. Each DDNTP is labeled with a unique fluorescent color, like A for green, T for red, G for black or yellow, and C for blue. The result is a mixture of fragments of different lengths, each ending with a known base. When these fragments are separated by size using capillary electrophoresis, a fluorescent signal appears in order, revealing the DNA sequence. This method remains the gold standard for accuracy, still used today to verify clone sequences. Next, let's talk about next generation sequencing, or NGS. Modern technology has scaled Sanger's idea to an unimaginable level. In next generation sequencing, millions of fragments are sequenced simultaneously using a method called sequencing by synthesis. Each base added during synthesis emits a small flash of light recorded by a camera and then converted into digital data. In a single run, a sequencer can generate billions of base pairs of information, the equivalent of hundreds of genomes, in just a few hours. This revolutionized genomics, making large-scale projects like the Human Genome Project possible in a fraction of the time and cost once required. Let's recap the chain of techniques. Let's gather the ideas that you've learned. Vectors carry foreign DNA into host cells. Restriction enzymes cut DNA at specific sequences. Ligase joins fragments into recombinant DNA. Phosphatases prevent unwanted religation. Transformation introduces plasmids into bacteria. Blue-white screening visually identifies successful clones. Restriction mapping and sequencing confirms DNA identity. Sanger sequencing, the classic accurate chain termination method. And then next-gen sequencing, rapid, high-throughput genome analysis. Every genetic experiment from vaccine design to gene therapy rests on this framework. And so, the story comes full circle. From the hydrogen bonds of the double helix to the bright flashes of a sequencing machine. Every step is a continuation of the same principle. Understanding life by reading its language. DNA is more than a molecule. It is a script, written in 4 letters, read by enzymes, and now deciphered by us. The tools of molecular biology allow humanity to not only observe life, but to gently rewrite it, to study disease, to grow crops, to explore evolution, and to understand ourselves. As you rest, imagine the quiet hum of a sequencing instrument, light flickering, letters appearing one by one. A, T, G, C. The timeless rhythm of biology continuing on, even in the stillness of night. And remember, everything we've learned, from DNA stability to RNA's flexibility, to the tools that cut and read the code, is all part of one vast conversation between chemistry and life. Good night, sleep well, and let the language of molecules keep whispering in your dreams.